Hello professors and doctors, my name is Dr. Ahmed Aref and I'm working at the Biological Science Department, School of Dentistry, MSA University, Cairo, Egypt. I have been working in liver cancer research for the last 10 years. Hepatocellular carcinoma is one of the most common cancers in the world with a global incidence of over than 600,000 new cases per year. In the United States, it is the fastest growing cancer in men. In Egypt, the burden of HCC has been increasing in the last 10 years due to high prevalence of HCV virus in Egypt. More than 10% of the Egyptian population has been exposed to HCV virus. There are several treatment options for HCC, such as tumor resection, liver transplantation, and local radiofrequency ablation. These services haven't improved the overall long-term survival. Therefore, we need new services to prevent the recurrence of this fatal disease. Immunotherapy represents a potential approach for eradicating the residual tumors. Several tumor-associated antigens have been identified, but little success has been achieved in the early clinical trials. So, the identification of new antigens may help to improve this approach. During my visit to Dr. Martin Gede's lab in the Long Beach VA Research Center in California, my colleagues Dr. G and Neil focused on studying the expression of 15 tumor-associated antigens considered useful for immunotherapy. We worked with 3 HCC cell lines, HepG2, Hep3P, and PLCBRF5. We used human lymphocytes as a negative control. We also examined non-cancerous tissue and HCC samples taken from autopsy samples. We screened for messenger RNA for these tumor-associated antigens using quantitative real-time PCR technique. The presence of the best expressed ones was confirmed using specific antibodies by using intracellular flow cytometric and immunofluorescence techniques. We found that HCC is a non-immunogenic tumor and failed to express many of the universal tumor-associated antigens, such as survivin, Williams tumor, cancer embryonic antigen, SSX2. So we next examined more specific HCC tumor antigens. Alpha-fetoprotein, aspartyl beta-hydroxylase, glypican-3, hepatocellular carcinoma antigen 519, which is also known as TPX2, hepatocellular carcinoma antigen 587 were investigated. With the exception of the last one, the other 4 tumor-associated antigens were highly expressed in the HCC cell lines and the cancer tissue, and not expressed in the normal tissue. Alpha-fetoprotein showed the most variable phenotype and was not expressed in all the clinical HCC samples. Aspartyl beta-hydroxylase was found to be highly displayed in the HCC specimen, but unexpectedly we found high amounts of its messenger RNA in the lymphocytes. We focused more with the novel antigen, hepatocellular carcinoma antigen 519. We found that this antigen was significantly elevated within the HCC samples when compared to the non-cancerous tissue and lymphocytes, both at messenger RNA level and protein levels. Using the CyBase and biomass databases, we designed and synthesized 2 peptides which combine to HLA-A2 major histocompatibility antigen. These peptides were pulsed to HLA-A2 dendritic cells overnight. These antigen-presenting cells were allowed to incubate with HLA-A2 positive cytotoxic T lymphocytes, which were then left for expansion. These cytotoxic T lymphocytes killed only HepG2 cell lines which is positive for both HLA-A2 and hepatocellular carcinoma antigen 519, and didn't kill the other two cell lines which are positive for hepatocellular carcinoma antigen 519 but negative for HLA-A2. So the results of this work shows that this novel antigen may be a promising antigen for HCC immunotherapy, and it can be further developed for clinical trials.